started thinking about how can we like what are the sort of specific and granular problems that we might try and address. And one big thing that came out was really the supply chain for reagents, over and above, for example, the supply chain for equipment. So I'll mention hardware and equipment very briefly in the presentation, but for the last 10 years, I've also worked around both open sharing of the designs to make lab equipment and also using things like 3D printing and maker spaces to kind of produce lab equipment cheaply around the world. So I have an interest in lab equipment, but the very strong message we got during a number of kind of conversations with colleagues, meeting people, running workshops, was that even if you can acquire the equipment, it's the ongoing cost of supplying the reagents for that equipment that's, or not, it's the ongoing challenge of having the reagents in place. It's not just a cost issue, although that obviously is a problem. But what often drives up the cost is that oftentimes the manufacturing of those reagents is not happening close to the work. So this is just a map of exportable laboratory reagents using some, well, it's a somewhat old data set now, but unfortunately they do not update it very frequently. So this is the most complete data set that we have currently. You can see the darker blue areas have a high level of export of reagents, which we're kind of using as a proxy for manufacturing. And so manufacturing is mostly happening in North America, Europe, and parts of Asia. China and India have a lot of activity, but the majority of the African continent and most of Latin America has reasonably limited manufacturing capability. And this is a problem because it means that stuff has to move from here to here, or here to here, or here to here. And for enzymes, that usually means it needs to move on a cold chain. So we're shipping like dry ice. So in an era where we're all trying to be more environmentally conscious, we are shipping solidified carbon dioxide around the world to try and deliver these reagents in a working state. Has anybody had the experience of enzymes arriving in their lab not in a working state because they've thawed out en route or got damaged? Anybody had that experience? Okay, a couple of people. Okay, others I think have been quite lucky then. good that your cold chain is working successfully. But we definitely had reports of challenges either with dry ice not being restocked or just issues with things getting stuck in customs for far too long. So this is a challenge, and there's a cost and complexity issue, but there's also a timing issue around how long it takes. So I'm fortunate, being based in the UK, that we have a lot of distribution and a lot of life sciences and biotech companies. So typically for very standard reagents that we use day to day, we would wait a maximum of 2 days maybe for something to be delivered. Our colleagues in Cameroon, 4 weeks was probably about the best that they could hope for most things. This is clearly going to vary country by country, so I'd be interested in your experiences. And also vary depending on exactly what you're ordering. But regardless of whether it's 2 weeks or 4 weeks, it's a huge difference from 2 days. And that means if you need to redesign your experiments, something isn't working in a kind of plant gene editing context, for example, you've realized that the DNA polymerase that you were using is not getting you sufficient volume of amount of amplicon yield to really assess if you've had successful gene editing. Maybe your primers just aren't working, you need to reorder them. Maybe your gRNAs aren't working, you need to reorder them. Every time you kind of iterate, that adds a kind of time penalty, which is really a geographical time penalty, right? So researchers who are working in areas with weaker supply chains are at a massive disadvantage in terms of the speed at which you can do research. Again, I'm sure many of you are very aware of these problems in your day-to-day work. So this import dependency is really the area where I need to say that I'm okay being recorded. There you go. Now you can see the top of the slide. I think every time someone comes in the waiting room, it shows the meeting controls again. So this high import dependency, we argue, go back, causes a number of problems. We've discussed some of them already. So one of the ways in which we might think about resolving this is to address the limited manufacturing capability. That is not necessarily going to solve all of the problems, but it's one approach to seeing if we can manage to kind of unlock some of this dependency. So we've done a number of market analysis reports. I'm just going to highlight one from Kenya. But of the researchers that we surveyed in Kenya, 90% talked about long waiting times, increasing their project costs, and diminishing interest, kind of diminishing their opportunities to move either their research or their kind of innovation and commercial ideas forward. There was definitely a kind of push to continue some of the activities to try and reduce these deficiencies. I put an old version of the slide. Anyway, but the other idea here was that one of the challenges is that you need to have trust that the stuff works, and so ordering from international suppliers often is seen as a mark of quality. So one of the challenges we have is addressing, if we move to a local manufacturing model, how do we make it a kind of a good fit in the market and producing stuff where the quality is really trusted by local researchers. But there's clearly demand for reagents, and so we're trying to kind of create this environment where we can try and get accessible and affordable reagents and support in place. And the idea then is that sort of positive feedback loop, so you can increase research productivity by reducing that time, that waiting time. And one would hope, although this is kind of out of our hands, right? If there's increased productivity, there's perhaps increased investment in research and kind of more innovation activity. This is not something that we can directly impact, because that would be governments and research funders and kind of other people who are moving financing around. But if that was true, then one could imagine there'd be an increased demand for the reagents and support, because more activity is happening, and so the cycle continues. So this is the kind of the dream. But even if that entire cycle is going to be either very slow, or perhaps we've made some incorrect assumptions, it's clear that there is an opportunity to create faster and more affordable supply chains for reagents that could accelerate and democratize NGS research, gene editing research, basically any kind of advanced biotechnology research, and overcome some of these constraints, like the high cost, the fragile cold chain, a lack of local technical support, the slow delivery and customs delays. And this is it's an issue that has been very long standing. This problem for molecular diagnostics got a big boost in awareness due to the COVID pandemic. And the sort of long tail of that interest has now resulted in much more attention on the general issue of the lab supplies market for research. And so this is a recent commentary by the Science for Africa Foundation, where they were really looking at how the lab supplies market is a challenge for African science. They're doing a number of surveys of lab suppliers. I believe that this month they have a meeting coming together completely about supply chain for science in Africa and what might be done to improve the situation. So it's definitely an area of interest. And so we've already highlighted some of the things that local production could bring. You could argue that maybe the best approach is just to talk to the bigger companies and encourage them to send stuff faster and drop their prices. And I say, let's do that as well. However, the reason that we're proposing a local production approach is because it has some additional benefits, we feel. So one objective is to bring skill development and capacity building for biomanufacturing and for recombinant protein expression, because these are general purpose skills that can not only be beneficial for research, but also for entrepreneurship and kind of thinking about innovation. And so by empowering scientists to conduct that high quality research into the protein and protein production space, we feel like that's in and of itself a valuable contribution. There is, generally speaking, a cost reduction because you take out the shipping and cold chain and customs cost. Even if international suppliers really drop their prices, there's like they have a ceiling which they can lower, but there's a base price that it's always going to cost to ship something from one place to another. So we've done quite a number of calculations in different countries, and we feel like for research use only reagents, it is possible to reduce the cost through local manufacturing. I'm saying research use only because actually if you look at the diagnostics market, it's a different matter. But that's because diagnostics, a huge amount of the cost is regulation. It doesn't matter whether you're locally producing it or elsewhere, it has to go through regulatory processes. There's a sort of self-sufficiency and an autonomy piece, which has really been pushed as a political agenda on the African continent, certainly around the idea of self-sufficiency for molecular diagnostics and for kind of molecular tools in the health space. I think we've seen a little bit less of that push in the agriculture space and in other areas like sort of research use. But as I mentioned, with the Science for Africa Foundation and other funders taking an interest, I think we're starting to see that coming around. It's a great way to collaborate and network, and hopefully give some innovation ideas that could be taken forward to commercialization. So the overall goal is that we want to increase resilience and autonomy through a distributed supply chain for molecular biology reagents. That's the idea. How are we going to do it? Okay, so we think in ways of increasing access to reagents as being at least 3 areas that all need to be addressed sort of simultaneously. So one of those is technology. So we need accessible product and manufacturing technologies. Even if you're not manufacturing, which would imply a whole pipeline and workflow for manufacturing, being able to design molecular biology reagent products that are better suited to, for example, shipping at ambient temperature, to not necessarily having to have very stable cold chain the entire time that are being shipped and stored. This can all contribute to sort of supporting this increased access, because we're not expecting every single lab to be making their own reagents. We need, once you've got the technology, it's kind of fine to publish a paper and whatever, but it also needs to actually get disseminated. And so we think a lot about building community and how we can create sustainable networks for knowledge exchange and distribution. And the work that Mark and Onesmus and the program here are doing is very much aligned with that. So we're excited to exchange ideas. And the third piece is that, as I mentioned, we're not expecting every lab to produce these things. And so how do we support mission-driven companies to deliver access at scale? And that's something that we've been doing slowly as a challenge. I'll provide one example later of a company that we helped support to set up. And we now have a number of WhatsApp groups that are available for technical questions and kind of small companies from many parts of the world. I think we have about a dozen companies on there from Latin America, Africa, and Asia that are at least nominally connected and can kind of share some of their protocols. We've seen for sure protocols transferred from Cameroon to Indonesia, now from Indonesia to Morocco. We've got a group in Brazil that applied for funding to go and transfer a protocol to a company in South Africa. So these are all companies sharing their knowledge as well. That's a sort of, I'd say this side is less developed, but it's something that I think is important. So how do we think about the building blocks of what needs to be in place? I'm going to talk a little bit about toolkits for DNA for the manufacturing. So if you're going to make proteins, you need the DNA that encodes for the proteins. You need some reagents to make reagents, so that's another thing. Training, an appropriate lab space to do this work. And we think a lot about how to kind of have an open approach to all of this. So for the majority of our technologies and tools that we develop, we do not apply for IP. And in fact, we go to great lengths to make sure that whatever we make is open source and openly accessible and can be used for whatever purpose, commercial or not, and can be redistributed freely. I'm going to talk about a project later, which is our most plant gene editing related one. That one does have a patent from the organization that originally developed the toolkit, or a patent pending. And so, I mean, I'm not including stuff in this talk about IP and kind of openness, but although we work openly, there are definitely contexts in which there are benefits to protecting IP for different strategic reasons. So when we're thinking about what we do in terms of IP, open science, et cetera, the goal is always access. And what of the many legal opportunities and toolkits, tools that we can use will deliver the best access. And that's going to differ depending on exactly what the technology is and what the opportunity is. So the first part of the presentation, though, will focus very much on stuff that's entirely available with no restrictions. And we think a bit about entrepreneurship and how we can hopefully support that, and how we can kind of make equitable collaborations and partnerships. Okay, so what do you need to do the local manufacturing? So I'm sure I do not need to explain too much to the biologists in the room about what you require in terms of DNA to make a protein, but you need the DNA that encodes the protein. So we have made a whole set of the standard molecular biology tools like DNA polymerases, ligases, reverse transcriptases available. We have over 60 currently in the toolkit and another 20 going through the synthesis and screening process now. So that's great, but you also need to be able to actually express these things mostly in bacteria. So we've been working primarily in E. coli. You'll see later on that we do have some yeast work coming on board. There's also other groups that are working on Bacillus subtilis and other kind of organisms. But we also need kind of regulatory sequences. So maybe we want to express it in a certain timing, a certain way. We need promoter sequences. We need tags to purify the enzyme as well. So we basically created these building blocks in collaboration with many other people who put this together, and an initiative at an initiative at Stanford called Free Genes that helped us synthesize everything. So we have around 300 interchangeable elements that you can combine. We also recognize that many people do not necessarily want to do the combining themselves. And so we also have a kind of core toolkit of ready to express plasmids for just the very, but we only have about 16 of those so far, and we're validating others moving forward. So the goal eventually is you can either, if you're not a person who does a lot of cloning, you can just get the plasmid ready to go. If you want to tailor it to your own requirements, you can get the toolkit and you can kind of move the blocks around. So this is what we've kind of put together. This is what we currently have available. We have the open enzyme collections. We have a lot of reporter genes as well. So fluorescent proteins, for example. I mentioned the ready to express plasmids. These are, well, we design them for molecular diagnostic protocols, but that means that there's a lot of DNA polymerases and other stuff in there, so very relevant. We have, as I mentioned, an open yeast collection and a yeast protein expression toolkit that is coming soon. Hopefully by the end of the year that will be put together and the paper published and made available. And we're always open to new suggestions and ideas. The DNA toolkit itself is in about 500 labs in 50 countries, but as any of you who have been on the training know, having the DNA in the freezer, it's not quite the same as having actually produced and used the reagents. So this is great, but we do actually need people to take the DNA out and put it into organisms and make things and try them out in the lab and really attempt to kind of scale up and see what's possible. So we've built a community since 2020 called Reclone. And this is the sort of global community for people that are using all the toolkits that's generating open source resources and having a sort of distributed collaboration network. There are three primary missions for Reclone, having regional reagent hubs where the DNA is accessible locally so that you don't have to pay every time to get it imported. We have a monthly community call and also a forum online so that people can ask questions or kind of share protocols. We have a repository of protocols on a site called protocols.io, but we're also building out open access education and training resources for all of the work that we do. Those who have taken the enzyme manufacturing masterclass, usually the presentations and the manuals are shared with the course participants afterwards. They can go and use them. They are, because we've been piloting the course to date, the final versions of that are not currently online, but they are going to be very soon, all under open licenses and available. And the Reclone team is basically led out of the UK, Brazil, and Chile at the moment. But we have reagent hubs. There's one, the furthest progressed is currently in Argentina, but we have one coming on board in Ghana and one in the Philippines. And these are sort of more the regional hubs, but then we're also building national points of contact as well. So if anybody is interested, let me know in terms of that. So we've built that network. There's probably about 100 researchers currently on the forum. There were around, I think, 30 to 40 people in Argentina last week for a workshop there, which is very exciting. But we've been fortunate enough so the Latin American Reclone group just received a 2-year grant from the Chan Zuckerberg Initiative. So that's enabled us to kind of run these activities. And we're now looking how we can kind of get similar funding for the African network and also the Asian network. So we're hopefully manage to do that soon and be able to follow suit. So we've got some DNA toolkits that in principle one can use to make molecular reagents, specifically enzymes. And we have a network that can spread the DNA and also in principle run training and other activities and provide a kind of technical and peer support network. But hands up who has done any recombinant protein expression outside of a training course that you might have done with me. So anyone who's using recombinant protein expression in their labs right now as a technique. Okay, this is very common. Anyone who knows of somebody in their institute or their university that's doing recombinant protein expression? Okay, a couple of people, great. Yeah, so it's not a technique that's as widespread as one might hope. And so firstly, we do need to do more training. Secondly, we want to make the kind of actual technology as accessible as possible for the manufacturing side. We want to kind of apply the same principles. The whole point of doing the local manufacturing is that you don't need to ship in enzymes, right? We're trying to get rid of the importation step. But if you then have to just import a whole bunch of specialized chemicals, it's not really like solving the problem, it's just moving the problem to another thing. So if the upstream requirements for manufacturing also involve a lot of stuff on cold chain and a lot of stuff that's hazardous and a lot of stuff that's difficult to ship, then we have issues. So one of the things that we've been doing is minimizing imports. One example is that we can induce the expression of our proteins using dried milk powder, which is available anywhere in the world in supermarkets. So typically this is done, the kind of canonical expression protocol is you use something called IPTG, which is a chemical, but it's basically a derivative of allolactose, which is itself a derivative of lactose. So you can basically use lactose. It has some disadvantages, but we use what's called an autoinduction protocol. I won't go through how autoinduction works, but please ask me afterwards if you're interested. The point is that we originally tried using fresh milk and whey, and then we tried milk powder, and everything works. So you can get a nice amount. This is on an SDS page gel open vent, which is a DNA polymerase used for PCR. You can see a nice big fat band. So we have protein, and it's working great. One of the challenges we did have is that this is what it looks like when you put powdered milk or fresh milk into a bacterial culture. And those of you who've done microbiology will see the obvious challenge here is that it's milky looking. So we don't actually know if this is actual milk or bacteria or contamination. It could be any of those three. And so this is not ideal for liquid culture. And so we had to problem solve a little bit more and thought, well, okay, we really want to use milk powder because it's super easy to get hold of and very cheap. But liquid culture is going to be a problem. Also, at the same time, we had collaborators who were really struggling with accessibility of shaking incubators. So typically, if you're making protein, you have massive, heavy incubators that shake these flasks, but they're expensive. They're very heavy to ship and they break because there's quite a lot of mechanical forces on them. And then when they break, you've got to fix them. It takes time. So that wasn't ideal. The other thing we found was that oftentimes, if you're doing recombinant protein expression, you have big centrifuges so that you can take liters potentially of culture and spin it all down at once. And very few labs that we were working with had large scale centrifuges. In fact, many just had your standard benchtop centrifuges. So they could only do Eppendorf tubes or like maybe a 50 ml Vulcan if they were lucky, which is very tedious when you've got to dispense all of your protein solution. So then we moved instead to do all of this the same, but on Petri dishes, because E. coli also grows on a Petri dish. So we just did the media, but with agar. We get a lovely rich lawn of bacteria there. This is on, oh, the video is buffering, sorry. I think I'm on Edgerome, but it's slow for whatever reason. Yeah, that's LB media, not so good. The one before was a very rich media. And so we discard the LB one, but in a second, you'll see how easy it is to harvest all of this biomass. So what has happened is the night before, someone's streaked this plate, left it overnight in the incubator, come back in the morning. We now use glass slides for this because it's safer, but you can see it scrapes off really nicely, and then you can just put that straight into your tube, no centrifugation required, and you can go forward and process these cells to make your enzymes. So these ones are expressing, again, open vent. And you can see you get a bit more in the liquid culture, but there's plenty on your plates as well. I mean, you don't need much of this. So one of those little standard Petri dishes will do thousands of PCR reactions worth of enzyme. So the yield is pretty good. And we're not really in the business of absolutely maximizing the yield. We just need to get a useful amount. so we were pretty happy with this. And this is now basically our standard kind of expression protocol. It makes life much easier for training because very few labs where we do the training have the necessary shaking incubators and big centrifuges. So now we can take this on the road, but we can also scale it up as well. So this is the protocol on protocols.io. If anyone's interested, you can just Google plate protein expression and you will find that available. And I thought I had put a picture in of us scaling it up. Okay, there may be one coming. I thought it was after the video, but it might be in a couple of slides' time. Okay, so number one is reduce the imports. Milk powder, no fancy chemicals. Number two, we want the biology to do most of the work for us, ideally. So we just saw an SDS page gel. Whose lab has SDS page set up for running proteins on gels? Okay, 1, 2. Funnily enough, I think it's the same people that had recombinant protein expression in their institutes, which makes a lot of sense. But this is, again, a technique that is not set up in the majority of molecular labs that we've worked with. So we don't really want to be running a gel every time we want to know, has our protein expressed? It's very useful to run a gel for quality control purposes, and especially when you're like developing the technique and just making sure that everything's fine because you can see the size of the protein, etc. But once the protocol is all set up, we're confident that we're expressing the right thing. We followed suit with a colleague, Professor Lisa Hall, took inspiration from her work, where she was testing the attachment of fluorescent proteins directly to enzymes as a kind of like built-in quality check. So now there are some proteins you cannot do this for, but our experience has been that the majority of the kind of workhorse molecular biology enzymes, you can put a fluorescent protein attached to it and it still works. There may be a slight drop-off in activity, but generally not so much that it's a problem, and you just adjust for that when you're doing your unit activity calculations. So the great thing about this is before the culture, after the culture. If we didn't have this, then before and after would look exactly the same, and we have no idea if we have protein or not. But with the built-in reporter, we can check. You could even, in principle, kind of calibrate how much expression you're expecting before you want to harvest, if you want to do these types of experiments. So again, it just cuts out an entire part of the pipeline and makes life a lot easier. And also it's pretty colors. So as biologists, we always love a bit of pretty color. Also works on plates. So you can see very obviously here the difference between the expression and not expression. This is a course that we ran in Senegal, and you can see we've adapted to use these glass slides now instead of the razor blades because they're quite dangerous. And this is what it looks like when you've scraped off the material, and it's just resuspended there in a little bit of PBS. And we did in this case run the SDS page, and as we expected, you can see expression, but we knew that because it's pink, so nice and easy. This is the one about scaling up that I thought was after the video for some reason. So yeah, how do we make this bigger? It's a bit annoying to do loads and loads of little Petri dishes. Well, you can just do big Petri dishes. In fact, these aren't Petri dishes. These are A4 paper holders, which we bought for like €3 each, and they're autoclavable, so you can keep using them. We've autoclaved them at least 20 times. They may well be perpetually fine to be autoclaved, we don't know. At some point they're going to break down, but they're polypropylene. You stick them in the autoclave and you can keep reusing them as much as you want, and then you can get quite a lot of protein from these different. This is sort of, in this case, we've been using them to express different proteins, but we're currently running an experiment to see if we stack 6 of them in a very reasonably priced small incubator, which costs about €150, how many PCR reactions can we make with that? Our like lower estimate, which we'd be disappointed if we didn't hit, is 100,000 PCR reactions. I'm hoping for more, but you can imagine that's quite a lot. That's probably more than a year's worth of PCR reactions for certainly many labs and potentially even some institutes or departments. So in one go, in one kind of miniature sized incubator with frankly, very little effort. And so this is the sort of technology that we'd really like people to just be able to use. So we can clearly express the proteins. There's now the topic of purification. So this is a little bit more complicated, but we should also always question our assumptions. And we have some collaborators in the US at University of Texas, Austin, Professor Andy Ellington and Dr. Sanchita Bhadra. And they published in 2018, so I'm guessing around 2015-16, they were asking themselves, but do we really need to purify the enzymes? Like maybe we're just making extra work for ourselves. And so they have a very nice protocol called cellular reagents, which they published in 2018, and then we subsequently worked with them to kind of even lower the cost and complexity of their protocol even more. And so we have some joint publications in that area. But basically here, we get our DNA fragments, we put them together. So referencing our previous slides, we put them in our E. coli, and then we have transcription and translation. We get our protein, and basically we just leave it in there. So we leave the protein in the cells, we collect the cells, which we've just seen, scrape them off the plate. We wash them in buffer just to kind of clear off any of the any sort of debris or any of the media. And then we basically dry them down just in the cells. And because they're kind of packaged in there, they're actually protected. And you can store these for at least 7 months at room temperature. And then when you rehydrate them, the PCR will work. So these are no purification needed. This is what we teach on our training courses as a starting point. So the initial training course really teaches protein expression and then basically creation of the cellular reagents. We're working on an advanced course which will do actual purification, but this works very well. So this is just another version of the protocol. You can see here we've measured the culture density, we've washed it, and then what happens is we dry them overnight. And no freeze dryer, anything like that. It's just a pot with some desiccant in it. So I think this is calcium carbonate. We use silica beads in my lab, but this is the Ellington Group's protocol. So any desiccant, I mean, stuff that you put in kind of packages to stop things getting too damp. It works even better and quicker if you use like a vacuum pump as well, which you can get. Usually most chemist labs have a kind of simple vacuum pump, or we found you can also, at least in Europe, get kind of vacuum packaging for food. And so you can get like very cheap sort of basically food containers, but with a vacuum seal so that you can put them under vacuum as well. We incubate them overnight at 37. You have to be careful how much liquid you put in because you really do want them to dehydrate overnight, so not too much liquid. And then in the morning, they look like this. And then when you want to use them, you just add your buffer and you're away. So you then use that like you would your enzyme from New England Biolabs or wherever you've got it from. I don't have a gel, but it does work. You can read the papers to see some pretty gels. I say whenever we've had courses where something hasn't worked, it's not been a result of the cellular reagents protocol not working. It's usually been a result of some mix-up with the cells or with the expression. So we have a very strong track record in this protocol working well, and we've replicated it in quite a number of countries now. That's what they've been learning in Argentina this week. So I think there's been people from Peru, Chile, Argentina, Brazil, I think Ecuador, maybe Colombia, have all been doing this practical over the last couple of days. Okay, so those are our kind of three of several ways. I didn't want to we're developing a bunch of other technologies as well, but those are just 3 examples of how we're trying to make it as easy as possible to do this work. I did mention I'll have a couple of slides on hardware. So measuring, for example, the fluorescence of your culture. I mean, you can eyeball it, it's pink, it's worked, tick. Maybe you want to be a little bit more accurate and quantitative. Maybe you want to test your polymerase activity. This usually requires some kind of fluorescent readout. There are ways in which we can do it cheaply with just gels. But we're trying to work on sort of projects where we can get small instruments that can also help do this type of work well and do the quality control well. And so Franquero has just published, it's still actually uncorrected proof, but it is technically published in PLOS Biology, this Qubite, which is an open source fluorimeter that fits 8 PCR tubes for an 8-strip. And the cost of the sort of materials for this is about €60, so it's very cheap. It's got a 3D printed heater block, a kind of electronics board underneath that he's designed, a 3D printed case, and it compares quite favorably to a commercial thermocycler for things like isothermal amplification. And so this is perfect for measuring, for example, the activity of our open vent. And I have a student, well, actually multiple students working over the summer on making sure that we can use this to test enzyme activity and have a very standardized protocol that's nice and easy. But he's published a whole bunch of other things you can do in this. He's got some CRISPR diagnostics experiments, so not CRISPR genome editing, but CRISPR for biosensing. He's got some cell-free protein expression and other things in the paper, many, many use cases. So we're quite excited about that. In a slightly more plant-related one, currently in my lab in Italy, we're building, well, we have a couple of these 3D printed microscopes. It's called the open flexoscope. They're about, well, depending on the setup that you do, like 120 to 250 euros. And we're now integrating fluorescence into those. So I say we're integrating, other people have developed a fluorescent system, which we're putting in so that we can look at protoplasts. So this is an image from one of these of protoplasts, and you can see them, which is good. I'm not sure how the contrast is here. This is just bright field. But our next goal is that we'll take the same images, but with GFP filters, so that we can test transient expression of GFP in the protoplasts without having a fancy kind of fluorescent microscope. And this is great. You can take it with you. You can pack it in your suitcase and travel wherever you need to do your experiments. It's not going to work for all sample types. And I'm not a plant person. I had no idea how big protoplasts were. But my postdoc Birhan sent me this image a couple of weeks ago. I was like, yes, I think this might work. So I actually have the optical filters with me, that we were waiting for them to be delivered. So now when I'm back again in Italy, I was supposed to leave them in Italy, basically. I forgot. I brought them to Belgium with me accidentally. so when I get back, we'll construct the fluorescent version of this microscope, and then hopefully we'll sort of have a nice lab in a box type of setup that we can take elsewhere. So hardware is exciting. This talk was not about hardware, but I really wanted to throw in just a couple of like interesting projects. We've run a lot of training. I mean, we've trained over 300 scientists. We set up a lab in Cameroon where we had a number of internships that we hosted, teaching people about the enzyme production. We've also co-supervised a couple of master's students. Actually, by now it's more than 2, but yeah, two that were sort of very formally co-supervised by me or somebody on the team. And so we really like running this training. We feel now like we're getting to the point where we have a kind of product training, like a one we can roll out for the basic work. And we're working now on kind of how to advance that towards plant engineering. So some of you may recognize yourselves in some of these photos from Kenya and Ethiopia. And this has been through a number of projects, I should say. So not just 1 or 2 kind of grants and projects and collaborations, but across many years of work with many partners. I'll talk a bit more about the Kenya and Ethiopia one in a second. This is just a very brief thing to say. I mentioned that we would like to support companies. The one that we've supported most so far is through a nonprofit called Beneficial Bio, and we supported a company called Maboa Lab to set up in Cameroon. And so this is the production lab that they set up for producing the enzymes, and they started selling to customers and had a product range. Cameroon ended up being quite a difficult market, and for a number of reasons, we now have shuttered this initiative. But I mean, on the plus side, we learned a lot, which we're translating to other areas. And all of the people in this photograph who were members of the team really benefited from their experience and have gone on to do other things. So Jan here he was our sales director, and he's off doing an MBA in Canada right now. Stefan is doing a PhD with me in Cambridge, and Minette, yes, Stefan is doing a PhD with me in Trieste in Italy, and Minette is doing a PhD in Cambridge, and all of the other team have gone on to do really well. So it ended well. We were able to make these products, for example, so gel loading dyes. These were all 100% manufactured in Cameroon, these ones. These ones we partnered with another manufacturing collaborator for that, but they were also making the master mixes in Cameroon too. And so, yeah, they actually were doing them with colored dyes as well, so direct loading master mixes. And we have all of the recipes for this on the protocols.io, so you can find the recipes there. Several people in the room have some experience of trying to do this as well. Anyway, okay, so that's reagents. The plant part, it's short, I promise, because I've been talking for a long time. So how is this applicable to plant gene editing, I hear you ask, because so far I've just talked about a lot of bacteria. Well, so we are partnering with the Growmore Foundation, who have put together, prior to us getting involved in the project, something that they call the Enable Toolkit. So this is Enable Gene Editing Implanter Toolkit. It's basically 5 plasmids that you can put together to produce a final plant gene editing construct for either transient or stable expression. And they've been working with Birhan Abati Edesi in my lab, who's working really on kind of validating the toolkit works. He's very excited because he's currently in Milan and he's got stably transformed Arabidopsis. Yes, he sent me a very happy photograph the other day. We'd already tested the protoplasts for the transient expression we're working in Trieste, so he's shown it works in both rice and also in the Arabidopsis, but we need to obviously analyze all of that data and write it up for the manuscript, which is in preparation. So for that reason, I'm not going to present the data because we don't have it in a final form yet, but just thought, it's working, which is great. And Growmore Foundation have been kind of developing this for a number of years, really with the idea of having this kind of easy to use toolkit for training, potentially also for research if people find that it's relevant to their particular plant requirements. So we have a kind of the DNA basically has been sorted. So if we remember that kind of structure diagram earlier in the presentation, we need some DNA, great, we have that. We also need the reagents, and that's where we're kind of working now to get an ambient temperature shippable molecular toolkit for the cloning part of putting together these guide RNA CRISPR constructs. So we need DNA polymerases. We've shown you that we can do those ones. We can do DNA ligases as well. The more challenging one is restriction endonucleases, because the cells are not happy with expressing proteins that chop up DNA. So you have one thing you can do is basically you have to express both the restriction enzyme and the methyltransferase that protects the DNA from the restriction enzyme. We have a couple of ideas about how we can express them without having the second gene, which we're trying at the moment. So the goal by the end of the year is to have tested both that we can, at least in the initial instance using commercial enzymes, that we can make, we can stabilize all of these proteins enough to ship out without having a cold chain and store them at room temperature for some time. And then ultimately that we can easily locally manufacture them, adopting the techniques that we've already been using, maybe with some slight modifications for the restriction enzymes. But yeah, so we're really open to thoughts on this. We're sort of setting out now on this adventure. And we have a whole set of training using the Enable Toolkit, which I'm very happy to share the kind of program of what that training looked like. We just did one pilot so far. Was anyone on the pilot in Ethiopia? Aha, Onesmus, yes. Okay, all right. Fair enough. I should have remembered Onesmus was a co-trainer on the pilot in Ethiopia. Yeah. So we did a test. There were many improvements that we need to make. That's why we're not releasing. So this training materials and everything will probably be released at the start of next year, because we're still working on making it better. We don't want to release something that's had some challenges, as Onesmus will vouch. But in general, it worked. We just want to make sure it's very high quality before we let other people loose with it. But there was a lot of really good feedback from that, and people enjoyed the opportunity. We're aware that we're basically training the molecular biology part of this, and I said I'm not a plant person, and I know from plant people that it's the plant transformation part that's the really, really tricky bit. So we're not solving all of the problems, but we hope at least to make the molecular bit a little bit easier. And this was part of a Kenyan Ethiopian project called Striga Smart Sorghum for Africa. And just to say that as part of that project, we were also thinking about developing a kind of innovation space. There's some training here. I definitely think there's people, Joshua's on here at least once, if not twice, on these photos. And so, as Joshua can tell you, this hasn't materialized yet, right? But we're still being worked on at Kenyatta. So we're hoping this sort of, maybe not looking exactly like the computer design, but the vision of having this AfriBio hub where we can do collaborative work and also have the reagent manufacturing set up as a kind of initiative to support related projects is something that's still very much a work in progress and still actively being moved forward by the many partners who are part of that project, who are here. This is us at the Sorghum Festival at the ABBC meeting, which I think, Mark, we met there. So this is the initiator of me being here. And look how much fun everybody's having, drinking. So the idea here was that everything was made with sorghum because we were looking at gene editing, an African-led gene editing project for striga-resistant sorghum, which if you want to know more about, ask Joshua, he can tell you all about striga. but the idea here was to show that, you know, if you can get striga-resistant sorghum and increase sorghum yields, there is downstream opportunity for value-added products. Great, so that's just telling me I need to stop. And so we were drinking sorghum beer and eating lots of baked goods with sorghum, etc., etc., etc. So it was a really nice evening. And yeah, it was also where I met Mark. So, so I think that's kind of it. Yeah, and the future is really, we want to keep building out all of this stuff. As I mentioned, my lab really only started working on the plant stuff. I'd say, when did Birhan join us? Like, basically, we've been on it for about 9 months, I would say. So we're pretty happy with how things are going so far, but we are very, very much open to suggestions. What will be most useful for your work? Where are the limitations that you're seeing? What are the things you're finding really difficult to get hold of? That will be useful information to know. And yeah, we're very, well, part of the reason for me being here is to also chat with Mark and Onesmus about, you know, how this intersects with the great work that they are doing, because, you know, there's a lot more to be covered. And I really see us as kind of hopefully providing the tools that many people providing training and kind of collaboration opportunities can make use of. This is just thanks to the whole team in Cambridge and Trieste and all of the various funders, but again, a massive thanks to Growmore Foundation, this is their logo, who really were the initiators of the Enable Toolkit, and they came to us and we kind of had a partnership discussion about how we could support the work they were doing and the challenges that they were seeing with rolling out training and implementation with this toolkit. But you know, the great thing about developing tools and technologies is that then it's not just for one initiative, right? Anybody can kind of use it for their own training. And I think one thing I've certainly found exploring this space is that there's so much potential in the plant gene editing area that, you know, if anything, we just need more people doing more stuff, right, to kind of spread these technologies. And so, yeah, get in touch if you're interested to collaborate. You can find me very easily online. We're always looking for people who might want to try out the protocols and feedback to us how they're working. We always have content development for various molecular biology, symbio courses and training, and yeah, a lot of different roles within the Reclone network as well. So thank you very much for your attention. I'm very open to questions if you have any, and yeah, your thoughts. Thank you very much. Yes. Yes. Mm-hmm. Mm-hmm. Yeah. So I think if they're wanting to, oh, sorry. Oh, they don't hear on things. Okay, so the question was, how are smaller companies that are trying to drive this market forward, specifically in places like Kenya, in East Africa? Yeah. Yeah. Okay, so companies that are not startups but have already been established or are kind of doing trying to expand into this manufacturing space. So those that we've spoken to who have an interest in taking this forward are generally very interested, because there's no, I mean, what they would get from us would basically be the DNA to produce whatever they want to produce, and then they can use the protocols if they want to, or develop their own if they want to. In this market, particularly for sort of standard PCR reagents, it's not an IP-driven market, it's a marketing-driven market. So you need to get the right price point and go and sell to people who will trust your product and buy your product. So I think it's not the sort of the idea of taking resources from other places. I haven't seen a lot of resistance to that. That's not necessarily true if they're trying to do, for example, a diagnostic test, maybe, because there would need is a different structure of resourcing. I can't. So we have not found a company in East Africa that has actively set up the manufacturing at a scale yet. We found a number of people, I mean, Joshua is one who's done some business pitching around this and has been trialing out the protocols. We found people who have, in principle, an interest. We're currently working with Kemri in Nairobi, who are in a sort of medical research center, but that's more that they already have a manufacturing suite for medical technologies. And so they're already established and already have a bunch of stuff. And we're working with them on getting the enzyme manufacturing set up. So if you know people, I'd be very interested to talk to them and find out how they receive it, because I don't think we've had those conversations with more established people. Mm-hmm. Yeah. 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 Yes. Yeah. 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 But they're currently distributors and they would want to be manufacturers. Is that the idea? Okay. Okay. Mm-hmm. Mm-hmm. Yeah, it's in process. So there were a number of partners putting that together. It is currently not set up yet. There were a number of issues with the space that was allocated for it. So, but certainly in the scoping, there was interest. I think running Kenya is a bit of a different situation. Our experience in Cameroon was there was a lot of demand for having a communal lab space, because many of the universities did not have molecular labs set up, particularly for master's students and kind of early stage PhD students, to be able to do projects. And so they appreciated a space which was affordable for them to come and do their experiments and have some technical support on hand and kind of support. And so people were coming from multiple universities, certainly across Yaoundé, and also sometimes from further afield to come. So I think it depends. We were not the people scoping the stakeholder work. For our role within that project was designing the lab and kind of recommending how to set up processes. So I can't point you to a stakeholder analysis document, but certainly our partners had spoken to a lot of other people about potential applications. So one would hope. For the manufacturing side, as I have not yet encountered a distributor who's in a serious way thinking about setting up manufacturing in East Africa. I've spoken to very large established international companies who have thought about it and not done it for various reasons. But I think those are solvable by having some various pipelines and other stuff in place. I think so I can give a couple of examples, just not East African, of small companies that are working with our materials. So there is a company in Indonesia, for example, that are manufacturing enzymes and also oligonucleotide primers. And so Stefan, who was running the initiative in Cameroon, went across to teach them the protocols for how to make enzymes. And they're now producing kits themselves. And they also got a big grant to set up this oligonucleotide manufacturing facility. They're now helping advise a group in Morocco that are trying to set up an oligonucleotide manufacturing facility. So, they're just kind of doing their thing. They receive the materials and some training, and then they're off and doing what they want to do. There's a company in Chile who received some DNA from the Reclone initiative that is currently manufacturing reagents for NGS. And, it's great. They're doing very well. They're a newish company, so but they've got some investment, they have some customers, things are going. And there's groups in South Africa as well that have took DNA from us and also used some of the protocols, although I think they also developed their own, that are making food testing kits and other kind of not medical but other things. So we know of at least, and also a company in Brazil that prototyped some production with the strains, and all of them were very happy to get the plasmids without having to pay, without having IP restrictions, without having other challenges. The nice thing about the local manufacturing is that they're not competing with each other, because their goal is not to be international distributors around the entire world. Their goal is to solve the problem by being inside the customs kind of boundary and producing enough for the local market. So it's not a problem for the producer in Brazil to share a protocol with the producer in South Africa, or the producer from Cameroon to share with Indonesia, because they're not competing with each other. So that's part of the reason we're trying to form this network, because it kind of de-risks. They're small companies, they can't do R&D on everything. So if they're all doing kind of little bits but they're willing to share, that really helps everybody out. And there are of course like new and improved PCR and qPCR products, but the majority of people are using enzymes that have not been patent, like their patent expired decades ago. So again, it's not, at least at that kind of basic molecular biology level, it's not a very IP driven field. There's no sort of like trade secrets or anything there. It's more about know-how to produce and manufacture well, and actually be able to do the business piece and do the selling of the product. And getting both of those things together is, I think, what we've seen is challenging. The technology is not so much of an issue. Like you can do the production, but even if you've got a freezer or fridge full of polymerase, you still need to get out and sell it. That's actually part of the kind of issue, because we're training scientists historically, and so scientists are maybe not the best salespeople either. So there's they need to kind of match up with the business people and have some investment and have some business support as well. And that's the piece that we haven't been doing specifically. But I think there's a lot of scope in that area to put together a more kind of fully featured support package. Which was something that was intended through SSSFA, but that project was cancelled early for reasons that I'm sure we're all very familiar with in the funding landscape at the moment. So. Mm-hmm. Mm-hmm. Yeah, absolutely. You need both. Yeah, all of them. They don't get the tag because also if we organize a practicum here, we have to take our tag from our lab based on the projects. So for a teacher, a lecturer, it's not always easy. So could those people here get access somehow to those kits? And how could we manage? Yeah, so what we've been doing so far is running the train. So the kits, we're still developing the kits to then find a manufacturer who will make the kits that can be shipped at ambient temperature, ideally directly to a local manufacturer who can make it. But as I just mentioned, that manufacturer then has to build out the business case and support everything else, which is challenging. So right now, our primary kind of people that we're working with are those that want to actually make the stuff in their own labs and trying to make that as easy as possible. And so we have the plasmids and cell strains. Typically what we've been doing is going somewhere, training locally, leaving behind the cell strains and the plasmids, and then they're accessible kind of in country for people to take and sort of set up in their own labs. What I think the main limitation is that after the course, people do need to have time to kind of implement the training in the different areas. And it's usually easier for people to do it in the institute we've run the training, because obviously we've kind of we've checked that all of the equipment is available. If it's not that institute, they have to go back and figure out what they've got. So for sure, if you're interested in this, what we can work on getting you either from people who already in your country have the DNA and cell lines, or through kind of figuring out a shipment, is the kind of plasmids and the bacteria for trying to make the DNA polymerases using that easy protocol, or try and put you in touch with somebody that we know who can already do that and then can supply it to you. What we're trying to explore at the minute, particularly for the plant gene editing toolkits that I mentioned, that's a test case basically for having a kind of ready to go kit that would have everything packaged as an actual product that we can ship out. The idea is initially we'll manufacture a pilot batch at ICGB, because we have all the stuff that we would need to do that, but then eventually we'd be looking to transfer that technology to some local manufacturers. So it's early stage, but if either of those things looks good to you, like you'd like to try, for example, making the cellular reagents, then I don't know what the easiest way to collect interest is. You can for sure drop me an email, and we will keep you in touch with any courses that are running, because it is helpful to come and actually be shown how to do it, or put you in touch with somebody that we know who's been on a previous course and can potentially help you, and also maybe put you on the waiting list when we've got some test kits that we want to ship out and have tested. Yeah, so I think we're at an early stage with some of that, but that's been our primary mechanism, is run a training course and then distribute. We are hoping to run at least one repeat of the plant gene editing molecular course before the end of the year, or possibly early next year. We don't know what country that's going to be in yet. So if you just drop me a note and say that you're interested, and we'll make sure that you're informed when that's happening, because we will be opening it to other people to come along, and then you can learn yourself how to do some of this stuff hands-on. Great. Yes, excellent. They were very good students on the course as well. Yeah. Yeah. Mm-hmm. Absolutely. We've been working quite extensively with Café, who's between. Oh, repeat the question. Okay, so the question was about at Addis Ababa University specifically, there's a challenge, and in Ethiopia specifically, there's a challenge around hard currency to buy reagents on the international market. So exchanging birr for USD is not easy. So how do we can we use local manufacturing as a workaround for that? I mean, for sure, if your input is significantly less expensive than the sort of end product. And yeah, it's something we've discussed quite a lot with Betin and Kasahun Tesfaye, who's also at AAU. Yeah, 100%. And we've delivered quite a bit of training in Ethiopia. So there are definitely people that have the capability, including your two students, to make this. And I think the bottleneck is then like taking that next step from the training to actually making and trying to build a business out of it. And I think one of the challenges that I mentioned before is that we're selecting people to be on our training courses because of their scientific research interests. And that doesn't necessarily mean that those people are the same people that want to go and set up a joint venture or a business and do all of the extra stuff. So I would say if you want to do this for real, then you need to assemble a team of people who have already done the training, but I mean, they can be trained, who have the technical interest to actually manufacture stuff, which is different from being interested in researching stuff, right? So that's a different type of motivation, and who have the motivation to really do the business piece. And if you can find the technical and the business people and they're willing to really give it a go, then I think it could happen. If you don't have that, or it's kind of one person who's trying to do everything, it's really difficult. So, and I'm happy to chat more, because as I say, we have a long-standing kind of link in Addis. So for sure, I think it's a very interesting economic case as well for the local manufacturing, definitely. Yeah, give it a try. Just do it. I mean, in Argentina, for example, in Buenos Aires, Cecilia D'Alessio's group basically make reagents for the institute that she works at the University of Buenos Aires. So they don't sell it outside, but they manufacture it internally and distribute it. And she has a technician who makes the stuff, tests the quality, and out it goes, and people use it for their day-to-day research. And that's a great way to kind of like practice and kind of get with people who are not going to be very angry at you. I mean, even if you're sending it to your colleagues, you definitely want to be doing the quality controls. I'm just saying there's a little bit of a difference between your colleague who can come and like politely ask you to please make it work better versus a customer who's paid for it, who you don't know, who's probably going to be a little bit more difficult to deal with. But yeah, I think you can kind of step up, make some for your lab, then think about maybe distributing to your neighboring labs, and then kind of go from there. There's an opportunity for sure. Yes. Yeah, well, it's definitely possible if you also express the methyltransferase. The problem is with some of the restriction enzymes that we want to do, the methyltransferase has not been published. And so short of like identifying, I mean, we might be able to identify it from the genome, but some of these restriction enzymes were basically identified and commercialized by companies like NEB. So a lot of them do not have publications associated with them. It's quite difficult to identify even the sequence. Some of them have patents, and we've got the sequences from the patent, but but often just for the restriction enzyme, not for the methyltransferase. So we're trying to find a way that's more generic. What we've tried so far, which has worked to an extent, is periplasmic expression. So you just export the restriction enzyme into the periplasm, the bit between the inner and outer membrane of the E. coli. In principle, then it's separated from the genome, and it shouldn't chew it up. And we successfully used that also to produce nucleases like benzonase, which chews up everything. So, the idea was if you can put benzonase into the periplasm, 100% you can put enzymes in there. And there's nothing published in the literature, but we did find a patent that had tried it with 2 restriction enzymes and validated that it worked for them. But the issue with periplasmic expression is it's very protein specific, and you can quickly inadvertently overwhelm the secretion machinery into the periplasm, and then you've got potentially folded protein in your cytosol, and then it's going to start damaging the genome. So it will work, but that requires like quite high you've got to test a whole suite of periplasmic expression tags with each protein that you want to do, because it's not universal basically, and you've got to be quite careful with your expression technique. So we have another couple of ideas for like ways of packaging proteins within the cell. There's a bunch of people who work on like synthetic cell structures that have been finding cool ways to basically form these liquid phase separations within cells. And so we're going to try out some of their technologies to basically like express the protein in the cytosol, but then kind of target them to a liquid phase separated condensate, which you can then purify. But that's a new idea. We don't know if it will work. It's worth a shot, because that should be more universal. I mean, they've already demonstrated that you can use the same tags to put a whole bunch of different proteins isolated in the same way. So we're trying to kind of, we like that idea as well, because it's even more using the biology to like solve a number of problems for you, because once it's put it into these kind of packages within the cell, they're not inclusion bodies, they're liquid phase, but you can easily separate them out. So that's also like an easy purification method built in as well, which would be lovely. So this is why we always want the biology to do the hard work so that you have to do as little as possible. I'm hopeful. Well, for the periplasmic stuff, we should have an idea, I would say, within the next like 4 to 6 months, if that's a goer or not, for enough of the restriction enzymes to make it useful. The other work will probably only start in September. So I mean, like, it could be 6 months, could be a year, who knows, because it's always difficult to tell, right? It's research. So, but yeah, we're trying. So I'm hopeful that at least for the enzymes that we're targeting, we already have had some success with the periplasmic. So most likely for the initial toolkit, we'll just do that, and then work on a more generalizable method later, because the enable toolkit only uses 2 restriction enzymes, so it's quite confined, but that doesn't necessarily mean they're the same as what you might already be using in your labs for assembling plasmids. So, right. Yeah, you can remark on how easy or difficult you found. I think you're the person who spent the longest time trying different replicates of the training and everything. So maybe you can comment on some of the challenges that or not that you found. Mm. Mm-hmm. Mm-hmm. With the deep vent, yeah, it does that. It needs a specific buffer, that one. Yeah. So are you finding that the NEB thermopole buffer recipe is not working for you? The NEB thermopole buffer recipe, the one from the protocol, is that not working for you at the moment? Yeah, it's not working. Okay, well, let's chat, because so the buffer that for sure works is the thermopole buffer, and the NEB one always works. The homemade one sometimes doesn't work. Usually what we've found is it's because the pH meters are not calibrated, or the it's a tris. It has tris in it, and some pH meters don't deal well with tris. So that's another problem we had in a different lab. I'm not saying that your pH meter is wrong or miscalibrated, but that is something you should check. Yeah, we'll have a chat later about the buffer situation, and make sure we can we can resolve that one. Uh-huh. Oh, amazing. I'm so pleased that the 3D printers are getting used. Excellent. That's great. Yeah. And if anyone, as I say, I have an interest in hardware as well, but we did not have enough time to cover everything in one go. So if you have questions about the sort of, if you happen to work in a university that has an engineering department that might have some 3D printers, you can do a lot of very cool stuff for your lab pretty cheaply. Plastic is quite cheap compared to shipping in. And when, okay, so it's maybe not that exciting to print just a standard rack, but think about it, you can do whatever rack you want. You can have any size of any size tube hole, and whatever you want to customize for your experiment. You can also do it in very cool colors. So we've been making magnetic racks for, if anyone uses mag beads for DNA purification, that kind of thing. They're very expensive, but they're just plastic with magnets, and there's many designs to print those online. So we had a little mag rack factory going in my lab for a while, and probably if you added up, we probably spent about, I don't know, £5, well, I'll translate it into euros, like €7 per rack. But I mean, you could buy them like 150 to 200 euros per rack. So you don't have to make many of them before your printer has paid for itself, because you can get a pretty reasonable printer for about 300 euros now if you buy well, without shipping. And if you want a really fancy one with AI that tells you when your print's about to go wrong, then it's like €750. But it's still significantly cheap. Even I was surprised, and I like to think I keep an eye on 3D printing, but I hadn't priced up printers for ages, and now these new ones, they're so like inexpensive for what they actually do. So you can, and they're like 4 times faster, so you can make very cool stuff quite quickly. But do make friends with engineers. Also make friends with chemists. So we have been looking also at how to synthesize DNA stains, which are quite expensive. So there's a dye called Eva Green, which we picked because chemically it's quite easy. It's currently on patent until 2026. So we're sort of getting it prepared and then at a certain point, but it's only patented in the US and Europe, so you for sure can make it in Kenya, no worries. They've not patented in Kenya, so it's okay, as long as you don't try then to ship it and sell it in the US, you're good. But yeah, we bought about $500 worth of raw materials, and we made $500,000 worth of DNA stain, if you like scale up how much we would pay. So yeah, we're really getting, as scientists and biologists in particular, we are getting really ripped off on DNA stain. If you look at the kind of cost of the materials versus the stuff, and it's a nice dye, and you can use it for normal PCR and qPCR. So we've just done another synthesis with our friendly organic synthetic chemist next door lab. Very handy, make friends with chemists. And we've taken photographs and everything of the protocol, so we'll be publishing that soon, so other people can have a go. And yeah, he said it's like a master's student in chemistry can make this stuff. It's not like you don't need a sort of expert to do it. So yeah. Mm-hmm. Mm-hmm. Yeah, it lasts for ages. I mean, that protocol, our colleagues in Latin America have been teaching that one to high school students so that they can do PCR practicals in the high schools as well. And the high school students the high school teachers, have found it really, really useful, because again, the main issue for them is just the cost of these particular reagents. The thing is, every time you go down a level, you always find something else that you need. So the one downside is you still currently need DNTPs for the master mix, which are not the most expensive component, but they're still a challenge. But there is a publication of a PhD student that I co-supervised who was looking at breaking down DNA into DNTPs, and she did show that you could use it for PCR, but there's a whole nother probably PhD or two to actually get that pipeline kind of finalized. So there always are a few caveats on when you start looking towards the master mix side, but yeah, you can. And so to the point, if you have the protocol and you want to have a go, just message me, because we have WhatsApp groups with all the people that have like done this before. And so the idea is you can just, if something goes wrong, just take a picture, send it to the WhatsApp group, and someone will help you figure out what went wrong. I think people are quite shy. So typically speaking, we don't get so many messages sent, but if someone does send a message, people are very helpful. so really don't be shy. Because yeah, exactly. And we have the online forum as well, but we feel like the WhatsApp groups, I think people feel a little bit less shy in the WhatsApp group compared to the online forum. But yeah, don't be shy. If you genuinely want to try these protocols out, let me put you on the WhatsApp group, and then if it's not pink, send us a photograph and say it's not pink, and we'll try and like troubleshoot with you why it's not pink. And that's part of the power of this community aspect as well, is that you're not stuck in a lab on your own being the only person that's doing it. There's people in labs all over the world that have probably encountered exactly the same problem and can help you out. Yeah. Yes. Mm. Mm-hmm. I do not have a slide, but yeah, I mean, basically, if you currently do not do so much molecular biology in your lab, and you're hoping to use your skills from this course and from the what you've learned with the mobile lab going forward, don't get stuck on the mode of you have to get the kind of commercial sort of full scale machinery for all of the stuff. There's often quite inexpensive options, usually that have been developed for educational purposes, but they're actually fine for research. So yeah, the mini PCR is one example. A little one, it's if you just search for mini PCR, you'll see them. It's about $750. It only does 16 wells. But we've been using one in my lab since 2018 and it's still going strong, and we've taken that in suitcases all over the place. It's still working fine, so it's robust. It's very nice. You can control it with your phone, so it's kind of, you know, it's got some nice touches. And there's a number of options that you can do. You can set up, as Asma said, you don't have to spend, you know, tens of thousands of euros to begin with. You can kind of but having said that, it's always a challenge, right? Even, you know, sometimes it doesn't really matter if it's €1,000 or 10,000. If you don't have €1,000, you still are gonna have issues. But yeah, definitely have a look around. And you can try the Reclone forum or the WhatsApp groups. Also the GOSH, Gathering for Open Science Hardware Forum. There's a lot of people on there who've looked into low-cost instrumentation, both commercial and kind of DIY build your own. And increasingly people selling kits. So the microscope that I showed you, for example, we've been building that in the lab, but you can just buy it. There are companies now that will sell you that 3D printed microscope, and they'll even customize it for you. Obviously, it's a bit more expensive because you're paying for their time. But you know, it's still much more affordable than other options. So yeah, we're very excited. We've just ordered a couple of a colorimeter and a fluorometer from a company called IO Rodeo that were like $200 each, and we're going to benchmark them against our commercial ones so that we can do more quality control with a sort of, you know, cuvette-based colorimeter and then a sort of PCR tube fluorometer as well. And yes, there's people are working on the equipment issue. One more. Yeah. Mm-hmm. Yeah, I'll give or take. So we in terms of setting up, we've set up a lab in Ghana and also one in Cameroon. And in terms of getting the basic equipment and the reagents, so from scratch, I mean, these were not even labs. So we had to put the benches in and do that sort of thing. I'm not going to include refurbishment costs in the estimate, but typically speaking, we'd be estimating a sort of small setup, probably around €30,000 would be fine. If you want to do something that's a bit, and if there's maybe also some refurbishment and also some more fancy equipment, you can do quite a lot for €50,000. But if you just want to start making the other things with all of the stuff where you don't need the big centrifuge and you've already got a molecular biology lab, the idea is that we're trying to do protocols that a person already has a standard molecular biology lab could do most of it. So if you already have a lab and equipment, I think based on our ordering of stuff, so for most of the training courses, we generally account for about 10 to 15,000 euros, because for sure there's going to be small bits of equipment that are not there. And so we normally have to put those in. And then we're usually buying everything from like from glassware through to kind of, you know, freezer, icebox things. Again, if you have a fully equipped molecular biology lab where you have all of that stuff in place, for the actual reagents to get started, it's not very expensive to get going. So, I mean, I think for a lot of it, again, it really depends on how much you already have. But our goal is to get to the point where we can send like a biomanufacturing in a box kit to a lab that they can open it and just get going with these protocols. And we're trying to price point that at around about 1,000 to 1,500 euros. But that's like everything, but it does assume that you have glassware, you have like a centrifuge, you have things like that, which, you know, for some people sounds very cheap, for some people still sounds very expensive. So it really depends on context if that's a good price point or not. And we're, as I say, we're trying to like figure out what's the sweet spot. So I have a team of students, masters. They're doing their MPhil in biotechnology. So they're working over the summer to kind of cost out this biomanufacturing in a box kit. What would we actually have to put in it? How much would that cost? What do people typically need? So I'll have a better idea on that piece before, but for sure, we've typically spent for a training course anywhere between 10 and 15,000 euros on a mixture of reagents, consumables, etc., including shipping and customs charges, which can, as you know, be very high. And then your 30 to 50,000 euros on setting up a lab. Yeah. But you can go as high as you want. I mean, you want some fancier stuff. Very welcome. Thank you very much.